My name's Steven Petrou, and I run a laboratory that really is focused on trying to change the lives of people with neurogenetic disorders. And neurogenetic diseases are a big cause of mortality and morbidity in our patients, and many cases there is no clinical option, so we don't have any therapies for these people. And so we've sort of taken it upon ourselves to try and do something about this. And what I'll show you tonight is one of our programs where we've made some inroads, talk a little bit about the importance of modelling, why we model, disease model, some of the advances we've made, and I'll end with a little bit of a technological development that's occurred in the lab that we hope will sort of progress and inform some of our next generation projects that we're looking at. So one of the first questions, we called it disease, brain in a dish, disease in a dish, and it's an emerging meaning. Quite recently, some of the advances you may have heard in stem cell technology are really fundamentally changing the way that we can investigate disease. But prior to that, scientists have been modelling diseases for years, and the model has been almost a rate-limiting step. How can you produce a system that lets you study it, tease it apart, prod it in a way, and learn from that in a way that makes sense to the human condition? If you're focused on delivering therapies, you need a system that when you study it, it tells you about the same mechanisms of disease that occur in a person. If you're testing a new drug or a new therapy on that model, you hope that what you learn from the model will predict what happens in the person. And that's a big gap now in the drug discovery world. There's many companies trying to develop therapies for hosts of disease, and the big problem is that we can do things in a model, and then as soon as we try to translate that and do a clinical trial, you get a lot of failures. The stroke world, as our director, Professor Jeffrey Donnan, can lament to you on other occasions, has done very poorly in this regard. There's been literally hundreds of studies that have been done in mouse models and other model systems, and not one of them, not one, has translated into a change in the way we deliver therapy and outcomes for strokes in human. And that's really disappointing, and it underlines a couple of issues that we have. One is on how well the experiments are performed, and that's something that the scientific community is currently trying to wrestle with, and measures are being taken now to try and improve that. But more fundamental to that is how good are the models that we study in the first place, and do we have a chance of those models actually translating and doing something in people? And I think why we study neurogenetic disorders is because we start with knowledge that's been gained in the clinic. So we look at patients, we see in many cases, in some of the diseases we're studying now that are quite severe, the patients are normally children that present at birth or shortly after birth with very severe symptoms. They can often have epilepsy. There'll be noticeable movement disorders. There'll be feeding disorders. And through genetic investigation, it's nowadays relatively affordable. It can cost as little as $1,000 or $2,000 to do a full sequencing in the child, to do a full sequencing in the parents, and to say, well, what's different? So what's changed in the genes of the parents and the kid? And you can see that with these new technologies, you can identify single changes in genes. And normally, there aren't that many changes that occur naturally. There's only 4 or 5 or 6 differences in the genes that make up our proteins that occur naturally, and that's a natural part of evolution, and it's called genetic drift. This is a natural process that occurs in all organisms and is a way that we adapt to our environment. There's always these very low levels of genetic variation introduced, and that's what drives evolution, survival of the fittest. These are the selfish gene idea that you may have heard or read about from Richard Dawkins. But when that happens, when that variation occurs, the wrong change, occurs in the wrong part of a gene, it can lead to disease. And this is what we're seeing, and now there's a revolution in genomic medicine nowadays that's occurred in Australia and around the globe. And we can now find the cause of these things, which just 10 years ago, a little as 10 years ago, we had no idea. These children would present and there'd be, there would be a lot of head scratching, there'd be syndrome naming, we'd be able to say there's a constellation of behavioural or phenotypic features in these kids, so you could give it a syndrome, but there's very little you could do, and we just a big question mark. And what's changed in the last 5 or 10 years is that genome sequencing has become very cheap, it's become widespread, you can access it, and now we know, for many of these cases, we know it's a mutation in gene X or gene Y, and we can quickly ascertain that that mutation wasn't in the parents. We can quickly model that in some sense. So how is the gene broken? We can express the gene, and that's been happening at a great level over the last 5 or 10 years. But it's also now presented a challenge with, well, we're getting more and more information about how genes can be broken, and there's all sorts of databases now appearing around the globe with lists of genes and where the mutations are. But we have to start consuming that information, that genetic information, and thinking, how do we do something now? We know how the genes are broken. Can we design therapies? Can we do things that, because of this knowledge of the actual way that the system is broken, can we fix it in a very directed or precise way? And you may have heard of this term called precision medicine. And precision medicine is the idea that if you have knowledge about the fundamental disease mechanism, well, then you can actually do a very targeted approach. If your car's broken and you know it's the spark plug, you put a new spark plug in. We used to think, well, the car doesn't move forward anyway. Well, I don't know why it's not moving forward. What do we do? But nowadays we know exactly what's wrong. And so it's incumbent upon us now to try and think about how to do something about that. A big aspect of that is how do we study these genes in the right contexts. A gene, and something like the brain, is a very complex organ. It's got lots, millions and millions and millions of neurons, that these neurons connect in myriads of ways. So a lot of the behaviour that you see is very difficult to predict. You can't, just by even having knowledge, if we build a map and I show someone this is how the brain is wired, we know every connection, we're still not going to really be able to look at that and say, oh, that's why it works. There's a branch of mathematics and physics called complex dynamical systems, and that tells you that knowledge of the underlying components doesn't necessarily tell you what the system does. And we've got that issue with the brain, so we need to be able to model it and to look at some of the emergent behaviour. So can we study things in the model that inform us of what's happening in the neurons, or in the brain, inform us of the disease process, or tell us something simple about whether a therapy that we're testing may or may not be effective. So it's important that we model, and we do it in epilepsy. And this is the reason that we do it in epilepsy. As you can see, probably 40 to 50% of all epilepsies have some genetic origin. It's unknown, but we're probably very confident that there's a huge genetic component. Whether or not there are some forms of epilepsy that you see that pass down in families, that's inherited, it's not such a major mutation or a change that you can't pass on your gene. And then those very, very severe cases that I was talking about that only occur in the children. And the reason you don't see those passing on, because these kids are so sick, they can't have families. Often, they live a very difficult life for 5 or 6 years and then they die. So those genes don't get passed on, so you don't see them as familial genes. The other mutations that you see that are inherited would have once arisen as a de novo mutation, and you call that a founder. And it could have been 300 or 400 years ago, a founder, this mutation occurred in this person, and then that then spread out through the population because it wasn't as severe enough that it actually stopped those individuals from procreating and having their own children. But we think there's a huge genetic component, and that work really started in Australia with the work of Sam Berkovic and Ingrid Scheffer at the Austin Hospital, and they really founded and globally founded this field of genetic epilepsy and really set us on this path, and that started in the mid-90s. And we are where we are today because of their pioneering efforts. This just gives you a rough idea of the number of Australians that will experience at least one seizure in their life. It's a big number. Epilepsy was a disorder that was often hidden because there was a lot of shame associated with it, and people didn't talk about whether there was epilepsy in the family. That's changed, thank goodness, in the recent years, but it's quite one of the most prevalent neurological disorders. Worldwide prevalence is massive. And the most disturbing thing is that at least 30 to 40% of people with epilepsy really don't have any effective drugs. So there's nothing we can give them that actually helps. Drugs, even over the last 30 to 40 years, there's been a lot of drug development. The drugs have got better insofar as you can take 1 a day versus 3 a day. You can take a small pill rather than a huge amount of drugs. All the side effects of toxicity, etc., are starting to be managed a little bit better, but we're not fundamentally changing the course of the disease. And there's 2 ways you can think about how you might treat a disease. You can treat the symptoms, or you can treat the cause. And if you treat the cause, there's a hope that you have this thing called disease-modifying therapy. That is, the way that you treat disease stops the evolution of that disease following a known path and giving rise to the actual condition. Most epilepsy drugs just cause a little bit of inhibition to parts of your brain, but they don't stop the seizures from actually generating internally. So it's a big problem. Now, what we're thinking is with all this new genetic knowledge and these very severe cases where we know it's a single gene causing the epilepsy, that if we can do something about those, if we can understand and tease apart those disorders, to deliver therapies in those, we hope to be able to tackle the broader patient population in future efforts. And if you look at it, you have a mutation in a gene, and you see a lot of different phenotypes or behaviours emerge in the patients. You can have cognitive impairment. You can have this horrible thing, this sudden unexpected death in epilepsy. 1% of people with epilepsy just die. We have no idea why, and that's a big number to be living with that 1% value. It is a horrible thing, and it's a big area of research now in understanding why this happens. Movement disorders you can see in some of the severe cases, and some therapies can try and tackle the seizures. Some therapies can try and tackle the movement disorders. Some therapies, and they could be physical or behavioural as well as drugs, may try and deal with the cognition. People are thinking of ways of dealing with the pseudo. But you can see once you've created a mutation, the genie's out of the bottle and it's wreaking havoc in many different systems. And if you're focusing on therapies that are patching up all these individual components, it's a really difficult task. And you might think, well, can we just go back one step and try and deal with the fundamental issue, which is the gene mutation? And if we deal with the mutation and somehow correct whatever that gene, it does, most genes, as you know, encode for proteins, and these proteins are the little things that do stuff in our bodies. These strings of amino acids that perform a range of different functions. And if we can fix that disorder, do all the other problems just get repaired without us having to individually deal and try to think of all the ways that these systems have broken. And that's where we're heading nowadays within. We think within the next 3 to 5 years, things that we're developing here at the Florey are going to be in the clinic around the globe, and we really think that we're going to have a chance of actually fundamentally changing the way we treat and deal with epilepsy. Now, the epilepsy I'm going to talk about today is these are the proteins that I mentioned. So this is a blown up picture of a cell. And the cell, as many of you may know, is made up of lipids, which are little fat molecules. And this is actually it was Benjamin Franklin who made the first accurate assessment of how thick a membrane would be by putting a drop of oil on a pond and seeing, well, that was a 1 milliliter drop of oil, how far did it spread? And he could just do a very simple calculation, and he gave a very close estimate to the thickness of a biological bilayer. And so what you're seeing here is the charged head of a little fat molecule, and this little bit is the little tail, a little carbon tail, like petrol. These little carbons here. And the carbons are very water-hating, called hydrophobic, and the heads are water-loving, and you have this thing called the hydrophobic effect, where all the fat likes to hang out together, and that's why they point together, they self-assemble. All the hydrophilic, water-loving surfaces love to point to where the water is, and that's why these membranes spontaneously form. So that's how you make a cell. And that's how cells evolved, because of the simple physical properties of the bilayer lipids. Now inside those things, what you can do now is you've got now this layer of lipids. It's pretty much tight. Nothing comes in, nothing goes out because of this little bilayer. Now you want to be able to talk to your world. You want to be able to signal. You want to see what's going on outside. Something's happened in the cell. How do I tell a neighbouring cell? One of the very early mechanisms that evolved to do that was these things called ion channels. They're called ion channels because they pass ions through them, potassium, sodium. These are the ions you find in seawater. The first organisms that were in seawater took advantage of the fact that you have plentiful supply of sodium ions, and there's lots of chloride in here. So basically you've got a little thing that can separate. Now, there's this particular type of transporter that actually will separate. So if you just encapsulated a piece of seawater, you might have high sodium on the inside and high sodium on the outside, exactly the same amount, because you've just taken a little sample of it. Now, if you want to be able to signal, it's really, really useful to separate those charges. This little special protein moves sodium out and potassium in. And you think, well, why do you want to do that? And when you do that, you now have changed the gradient. So you've got very high sodium outside, low sodium inside, but high potassium. So you've separated these two molecules. Now, if you make another protein that can allow these ions to move in under some sort of control, so it's a little tap, you can turn them off and on. And you can turn this tap off and on, and potassium will then move where it's lots of it from the inside and out, or you can turn this tap on, and sodium will move in because there's lots of it on the outside and not much on the inside. It'll just diffuse. And that simple mechanism allows you to now change the voltage across this cell. So as soon as you do this, you generate a voltage, and by opening and closing these channels in different ways, you actually can form electrical signaling. And this is the basis of signaling. These are the reasons muscles contract. These are the reasons neurons talk to one another. So these are the reasons the heart contracts. This is the basis of all the electricity in our body that we record when we put an ECG on the heart, an EEG on the brain, a myograph on the muscles. The reasons that we detect them is because of these little ion channels opening and closing. We see them in some of the earliest bacteria. They evolved. You see these very early types of ion channels that allowed signaling to occur. Now, because they're so fundamental, I mean, if I was to take a drug that blocked these sodium channels, I would be, out of it within seconds of taking it. There's absolutely, the real-time function that we do is completely controlled by these channels. So it's not as if you can block them and you'll function for 5 or 10 minutes. You'll completely be disabled by these. One of the puffertoxin, fugu, the Japanese delicacy that they worry about, because there's a toxin in the liver of that fish that blocks this particular sodium channel. So when you eat that toxin, rather than the sodium ion, this big fat toxin sits in there and doesn't let the sodium to go in or out, and that's why you die. So and then the fish evolved these mechanisms, and that'll become a little more relevant later because we've actually taken advantage of nature for one of the therapies we developed. The fish have evolved this mechanism, a way of disabling their prey. So ion channels are the real-time messengers of the body. And so it's almost no wonder you think, okay, if they're the things that are making our body work, our hearts work, our muscles work, there's no wonder that if you break one of these through genetic mutation, when you change your gene, you're changing the blueprint for the protein that you produce. And proteins are made of amino acids. There's 20 amino acids, like strings on a pearl. And the order which you assemble these amino acids, and sometimes some of these proteins might have 2,000 or 3,000 of these amino acids in a row, and they fold up into these complex structures. If one of them 1 out of the 2,000 or 3,000 is altered, you can completely disable this protein, or you can break it in very subtle ways. If it's broken too severely, well, that's not even compatible with life, and you don't even see that organism being, born or gestated. But if you break it subtly, you have disease. And that's what we're seeing now. Even though they're terrible diseases, the really, really bad mutations you don't even see. So you break it, and it's very easy to think how that might cause a brain disease. There are muscle diseases associated with mutations in ion channels. There are heart diseases, long QT syndrome, that some of you may have heard about, associated with mutations in ion channels. Epilepsies in particular are associated with changes in ion channels. So one of the ion channels live in nerves. And so these are nerves, and this just shows a point of contact where two nerves talk to one another. So nerves are avid communicators. They branch out. They have a part of the nerve that actually talks to other nerve. It's called the axon. And then the part of the other nerve that listens to that axon is called the dendrite. So there's a sending part and there's a listening part, every nerve. And they connect in very complex networks, and the architecture of those connections determines the function of the brain. And that complex orchestration of connections and different structures gives us the complexity. And the brain has evolved different parts. The hippocampus helps us store memory. The cerebellum helps us coordinate our walking. So we've put this sort of modular approach. These brains have got more and more complex. You can look in simple organisms, and all the nerves do is direct you towards light, or direct you towards, so that you sense. All organisms sense. They look at their environment, they process that information, and then they decide what to do. And you can almost look at that model for humans with complex social interactions and all the things we do. You can look at it in a single organism that's living in a pond, is trying to find a nutrient. If there's a sensor for the nutrient, and then it sends a signal to the muscle to turn in that direction. All of that's controlled by ion channels. And that same archetypal pattern is what drives us. It's driven by these nerves. They're full of different ion channels. And if one of those ion channels is broken in a funny way, you can give rise to too much electrical activity, which is what epilepsy is. Epilepsy is an electrical disorder of the brain that gives rise to aberrant firing of these neurons that produces the seizures and all the other things that we see in epilepsy. So then I said, we're going to look at severe genetic epilepsy, and this is one of the severe epilepsies. This one's actually called Dravet syndrome, but it doesn't really matter. What I'm going to show you is sort of a generic way that we can study these disorders. This one is rare. There's probably 1 in 30,000 people. But it's a huge burden on society. It's a terrible thing for these kids. A lot of these rare disorders fall under the radar for big drug companies to do something about because there's not enough of them. But the impact on families and carers is huge. These kids can live up until their 20s, 30s and older nowadays. And yet they need continual care. And so that we have to do something. And our idea is if we can just take one of these at a time and do something, then we've got a therapy now. Even if nothing else happens for 50 years or 100 years, we've got a therapy and we can tick that one off and move on to the next one. And the hope is in the next 40, 50 years that we've got a therapy for each of these conditions. There's only so many ways these genes are broken, so that's finite. We can deal with them all. Now these kids starts very early. They have epilepsy and seizures. They have intellectual disability. They don't move properly. Their stomachs and their autonomic system doesn't work well. And there's really urgent clinical needs. You may have heard of the cannabis trials, the cannabidiol trials that have been ongoing in Victoria around the world. Well, this epilepsy is one of the things they're trying to target with those. And there's some good news coming out with those, but it's not fundamentally changing the course of the disease. Cannabidiol might reduce the numbers of seizures these kids are having by 40 or 50%, but it's not making their intellectual disability better. It's not necessarily changing, so they're not all of a sudden taking this drug and now going off and kicking the football and doing the normal things that people want their kids to do. So we think we need to really rethink, and that's what we think we're going to do, rethink the way we tackle these disorders. So I said, often caused, in this case, that disease is caused by a mutation in an ion channel. And it's a specific ion channel in the brain And there are basically two types of nerve in the brain. There's those that are the excitatory nerves, like the accelerator in a car, and there are those that are the inhibitory nerves, like the brakes in a car. And as you know, to control a car, you need brakes and accelerators. So you need that control. Brains use a similar concept. We've got excitatory neurons, we have inhibitory neurons. Now, this particular mutation is in one of the neurons that controls the brakes. So the trouble in these kids, there's not enough braking, not enough inhibition. We can study it in various ways. This goes back to the modelling. One of the ways you can study it nowadays is you can take a tiny bit of skin and you can turn those skin cells into a stem cell. And this is the thing that's been in the news for a long time. 15 years ago, they thought the only way to have a stem cell was to actually take a little embryo, and that's why there was a lot of controversy around, well, you're killing something in order to have a tool in order to study. There's a huge amount of ethical dilemmas there. But around 10 years ago or so, a Japanese group discovered that you can actually take some skin and you can treat it in a certain way and you can actually make it go backwards. And when I did biology, I was told, well, you can never turn a cell back. Differentiation you go from a very primitive cell, and that changes and becomes a heart cell, or it becomes a brain cell, or it becomes a skin cell, and that's it. You can't go back. But I actually discovered you could turn it back. You could turn time backwards, you can treat it, and these cells get more and more primordial, and they become what we call pluripotent. That means they can become anything. So when the cell becomes in that pluripotent state, you start with a very complex cell like a fibroblast, you wind it all the way back and you make it this little baby cell, and then when it's in that state, you can then, with another series of treatments, turn it into a heart, turn it into a lung, turn it into a kidney. In fact, the first kidney in a dish was grown by Melissa Little's group at the Murdoch Children's here down the road. So Australia's in the thick of this. We're doing some fantastic things in science in Australia. So this process of taking a skin cell, turning it back to a primordial cell, and in this case, we can start to turn these into brain cells. So we can grow these in dishes, these cells and then you can start to see the electrical activity that is part of a normal way a brain cell functions. And so this really gives us an unprecedented opportunity to study human tissue, human models, and this person who we're studying may actually have the genetic disease that we're interested in. So we also get the genetic change in this person, so we have a really good way of studying the disorders now. And this has really only happened in the last 4 or 5 years that we're starting to do this. And we're poised in more complex ways. This forms a sheet of neurons. But now people are thinking, well, rather than a two-dimensional sheet, can we grow these into little balls? And they call those brainoids or organoids. So you can start to now introduce three-dimensional complexity into these structures because we want to model more complex behaviours. A sheet of cells connected, you may not be able to model more complex disease. If you want to model schizophrenia or cognitive disorders, you need more complexity in your model to be able to do that. And we're thinking by introducing more structural complexity, we can start to get the higher order functions emerging in these little organoids. What can we do with them? Well, we can put them on dishes, and here this measuring the activity in these things called micro or multi-electrode arrays. What they are is a substrate, glass or plastic, and about every 100 microns or 1/10 of a millimetre, you have this little electrode. And that can detect the electrical activity of the nerves that are near it. So these nerves are laid down, they self-assemble and connect to one another, and then when they actually are active, this electrode and this electrode and this electrode and this electrode will detect that activity. And that gives us now a way of saying, well, what's happening in this network? If you look at it, you don't see anything. You need a way, and this is a fairly non-invasive way, and the nice thing is we can have this in the dish and it can sit in there for weeks or months or longer. We can put a drug on, we can say, what does the drug do in the short term, or what does the drug do in the long term? So it gives us a lot of flexibility in how we we are studying these nerves, and the nice thing is they're human. So a big problem is you might make a drug that works in a mouse, as I said before, may not work in a human. A lot of that is because our pharmacology is different. And that's one thing, that the drug you make may interact differently with the human target than the mouse version of that same gene. But also, the way that our neurons are assembled are different to the mice. So the higher order things that emerge are different between mice and humans. And so we need ways of getting that complexity in there so that we can study those different behaviours. And what do you see? Well, this is one of the things. You may have seen EEGs before, brain waves. Well, this is sort of the dish version of that. This is one of those dishes that's got different electrodes, and these little blips are whenever those sodium channels I talked about open, they open and close briefly. And when they do that, they open, sodium goes in the cell, and then they close. And when you do that, the inside of the cell moves from being negative to being positive. And that's the fundamental way that neurons operate. They're called action potentials or spikes. That is the fundamental signalling process. We can see each of these blips up is recording that event from occurring when you see it occur. And this is from a patient that's got a mutation in an ion channel. This is a little American boy who we're working with. He was born. Within 2 or 3 hours of him being born, he was having his first seizure. He doesn't talk. He's like 3 years old now, and he's just starting to walk. He's got no verbal. He's got a lot of problems, and there's a huge amount of care goes into this kid. So I'm actually working with the parents, and my whole lab is obviously helping out enormously, working with the parents to try and think, can we fix this child's epilepsy? And if we can fix his, there's a whole bunch of other kids we know about, probably 5,000-6,000 around the globe that have the same type of genetic change that we can impact. And if you look at these, you can actually, these are the nerves playing back. And what you'll see, look at this. You can see the activity goes from being very quiescent to being really, really active. And then it quietens down again. You can see it happening here as well. And it goes quiet again. You wait a bit longer. See the activity slowly building again. It's building. And you can see some of the activity occurs in, this is one electrode, you can see here going really crazy again. And you can see, and this is one of those little dishes that's got multiple electrodes in it, and you can see that it's occurring simultaneously across multiple sites. So these neurons are talking to one another and entering this epileptic state. They're synchronizing their activity and they're firing in this uncontrolled. It's exactly what you see when you put EEGs on a real brain. So we think we're starting to get really good views now into we're asking the question, well, how far can you disassemble this brain and still get some behavior that makes sense? Because the more simple you can go, the more you can do. Because there's always this balance against, we can do some super duper complex experiment, but you can do that once every 2 months. But if you want to treat and develop drugs, you need hundreds or thousands of times of doing that experiment. So you need to simplify it as much as you can and still get the useful behaviour. This is one way of doing it. We can now do 24 of these at once. I didn't show it there, but we can buy these MEAs. This is a single one, but you can buy them here. And there's multiple. So now all of a sudden, from doing one experiment at a time, we can do 24. At the back, you can do 96 experiments at the same time. That extra capacity lets you broaden your net, lets you look for more therapeutic choices so that you've got a greater chance. Because a lot of drug discovery can be about just luck, which means that you need to cast a big net. We think what we're doing is less about luck and is more about design, but it's not always going to be the case. Now, if you look, as I mentioned before, in a normal person, we've got one set of genes from our mother, one set of genes from our father. We always have 2 copies. And some genetic disorders, such as cystic fibrosis, require that both copies be broken. So 1 in 25 people are carrying a mutation in the gene that gives you cystic fibrosis is called CFTR, or cystic fibrosis transmembrane conductance regulator. That gene is broken, and if you have 2 copies, you get the disease. In epilepsy, there are some conditions that you see that require 2 copies, but in these de novo cases, we have 2 copies, one from mum, one from dad, and they might be subtly different, but they're not disease-causing. You do see natural variation, and they're called polymorphisms in the population. There's some of these variations you see are as high as 1% of the population are different in a very subtle way, and we don't really know how they change the function of these genes, but these changes are important. It gives us diversity. It's why every person in this room looks different and is different, has different skills and different abilities in different areas. That's because of the diversity in the actual structure of these proteins. Now, as I said, in this particular epilepsy, one of them is rendered ineffective. It's the mutation, single mutation, breaks one of these. So rather than having, you need 2 copies in order to function properly. What happens in a normal, this is what, when we put an electrode on a neuron, this is what we can see. So this electrode lets us look inside the neuron to see what it's doing. And this is one of those neurons I said that causes the brain to be relaxed. It breaks, it's a brake, it's an inhibitor. And what you're doing here is you're telling the neuron, turn on a little bit, turn on more and more. So as we go here, we're instructing it to turn on more. It's like you're pushing the brake on your car harder and harder and harder. And as you push harder, you want the brakes to be applied harder. And you can see in the healthy neuron that's got 2 copies, you get that effect. But in the neuron from the patient, when you ask it to brake harder and harder, you see it starts to fail. There's not enough of there's enough of the sodium channel to support low levels of braking, but when you really call on it to do a lot of inhibition or braking, it starts to fail. So when a nerve's doing this down here, it's not actually talking to the other nerves. These nerves, when they do this, release a natural calming agent called GABA. That agent comes out and it calms nerves down, so it's the control that you have in the brain. When in these brains, they stop releasing GABA. It's like you've heard of VTAC and all those medical shows where the heart goes into this type of ventricular tachycardia. When it doesn't beat properly, it starts to vibrate, and then it can't function as a pump. Well, this is sort of a similar thing for a neuron. It doesn't fire these action potentials properly, can't release its GABA, you don't get enough inhibition, and you get rise to epilepsy. So we asked the question, well, if there's only 1 copy of these, not 2, and it's not, can we make this copy of the protein work harder? Excuse me. So can we make it work harder? And what we turned to was a colleague of mine in Queensland who studies venoms. So, why would you study venoms? Well, there's little creatures that, if you look at a venom is like a spider, when it bites you, it releases venom. A poison is that the animal could be poisoned. If you eat the whole animal, that's the poison. But the venom is the thing that it injects when it bites you. And there's all sorts of stuff in Queensland that does that. You walk along the beach, cone toxin, and these venoms are really complicated. They're not just one thing. They can have 400 peptides in them. We heard about that saga with Essendon kids receiving a peptide, and they're not innocuous things. I mean, these peptides are small. They're 34 amino acids, and some of these are the most potent biological agents that we know of. And so when you look at a spider venom, and this was a Tongan spider, it's got 400 or 500 of these. And Glenn's group is really good at separating the 400 or 500 into single components. You can take those things, and we know that a tiny little spider can knock out a full-grown person. So we know they're potent, and we know they're highly biological active. So we asked the question, well, is there any medical benefit to these venoms? So what you do, you can separate the venoms. We knew that this is the channel, the scientific number we give it, it's called SCN1A or NAV 1.1. It doesn't matter. And this is how, when you do an electrical experiment on this channel, and you tell it to turn on. These channels are really clever. They don't turn on for very long. You say on, and they go blip, and they turn off naturally. So they don't stay on. So even if you've got the signal, the light switch is on all the time, they just give a pulse. And you have to turn the light switch off in order to get that pulse again. So that's what they look like. They pulse. But what you can see here, where this is the amount of venom, 1 nanomolar. So a nanomolar is 1,000th of a millionth of a mole. So it's a very, very tiny amount of this venom. And as we started to increase the amount of venom, you can see that the pulse got fatter and fatter. So the question we asked is there an agent that can make this one healthy copy work more? Well, the answer seems to be yes. There is an agent. It's one of the components of a spider venom that does that. So at this level, that fulfilled our design criteria. This channel's broken. Can we make this one copy work harder? What does it do when you put it on those broken nerves that I told you before? So up the top is we put an electrode on one of those broken nerves, and you can see here, when we asked it to do some activity, it was okay. When we really asked it to do a lot, couldn't sustain the activity. It just gets tired and it runs out. This is exactly the same nerve on the bottom after we put the venom on. Completely maintains its firing. So we've been able to go, we can fix the protein, and now we can fix the nerve in exactly the same way we want. This now looks like a completely healthy neuron. Really important, but we only have one copy. We don't have 2 copies now. We only have one copy, one of them still broken. And importantly, when we turned to an animal model in this case, so we can genetically engineer mice. It sounds horrible, but we have to do this if we want to solve these diseases. We can genetically engineer the mice to have exactly the same genetic mutation that we see in people. And the mice display the epilepsy and all the problems that people do. If we give, at day 0, we had a group of mice that were untreated, and we had a group of mice that were treated. If you look at the untreated mice, they started having their seizures at day 0, having 4 seizures a day. By the second day, they were having 12 seizures a day. They were dead after that. Had really bad seizures and died. The animals that we treated started with 4. After day 1, they had 4. Day 2, they had 2 or 3 seizures. By day 4, they stopped having seizures. So we could show we can fix the broken ion channel, we can fix the neuron, and we can provide what we've seen in this preclinical model, one of the most effective therapies we've ever seen in this particular disorder. So we're happy to say that this has now been licensed to an American biotechnology company that we're heavily involved with at the Institute that is going to try and move this into people. And so we hope to see in several years, fundamental changes in the way that we are delivering care to these patients with Dravet syndrome. And so we're just delighted. We've actually got multiple programs now with different therapeutic, with gene therapy, so whatever it takes, we're looking at the genetic mechanism, we're studying in our disease in a dish models what the pathology is, we're thinking about a therapy approach that might work, and try and systematically go through as many of these particular genetic syndromes as we can. Before I end, I just want to talk about one of the other things that's a really important part of science is really that combination of technology and engineering and mathematics with the problems that you face when you're doing neuroscience and neurological research. So these things go in hand. It's a very interdisciplinary thing. And one of the big things, you may have all heard about MRI, which lets us look into the brains of people. We need to peer into these brains at much, much higher resolution than you can with MRI, but we need the advantages that you can rapidly scan an entire brain quickly and get very detailed information about how these neurons connect to one another, because in many diseases of the brain, these connections go awry. You can't see them at the level of the MRI, but we have done very detailed studies in time. This is a mouse brain, very tiny regions that take months and months of work to say, wow, there's changes in the way these neurons connect, but we need to understand the whole map. One of the new technologies that's emerging is that we can use genetic tools to make different neurons glow. So we can shine fluorescent light on them and they glow. And this is glowing green, some neurons are glowing red. But if you've got a little ball of fat, you can't see into it. So what we can do, and this is not in live, this is in dead brain imaging, but we can chemically treat these brains and make them transparent. So now you have a whole brain, it's transparent, and it's got these little filamentous neurons that are glowing with fluorescent colours. And how do you image this? We used to slice the brain apart into a thin piece, image that one piece, go back, take another slice, image that. Hugely laborious, and a year you can do a brain. And putting it together is just murderous. So is there another way to slice or section this brain that doesn't require physical destruction? Well, this technology that we and others are developing is called light sheet microscopes. What you do is, if you can imagine, if you've got this jug and it's clear, and if you're looking at it face on, there's all this information structure in it. If I could make a sheet of light that's thin as a piece of paper and just illuminate that one slice, when you look at it, all you're seeing is those neurons that are illuminated by that thin sheet of light. And then you can move that sheet back and forward, and as you move forward and back, you illuminate different sheets of information. And the neurons, because they're fluorescent, they're invisible until you shine this light on them. So you can imagine, if you can make a thin enough sheet of light, and you can control that sheet of light and control all the physical things that happen when you're imaging like this, you can start to use this method just to move this sheet of light through the actual brain. And I'll show you what some of the team members in my group have done. We're actually putting the sample here into this little chamber, and it's on the tip of this carrier is a tiny little piece of glass brain from a mouse. And these are the optics that produce the light sheet. And you can see the microscope that we've built. And we turn the lasers on, and we're shooting laser light down through this chamber, and we've produced, through these optics, producing a very, very thin sheet of light. Now if we zoom right in, you can see the sample here, and the nice thing is we've got a sheet of light, and we can move that sample back and forward through this sheet of light. And the team has built software, and you can see here how, as we move it, we reveal a different plane of image through that piece of the brain. You can see there's little dots of neurons here emerging. It was 5 or 10 years ago, it was almost impossible to do this with this speed. And it's an amazing, rapid use of technology. The big issues now relate to the software to deal with these massive images, how you reconstruct them, how you remove noise and other artifacts, and how you store massive amounts of data. And that's changing, the movement in that area is remarkable as well. Now this will show you what a view through the cerebellum, which is the part of the brain that controls movements, looks like. And you can see now the neurons flickering. You can see some of the axons from these neurons. Here you can see here axons. So this is just looking at it piece by piece. Then I think we put it together. Now this is after some image processing. So here we've got teams of physicists and engineers working with different biologists with different skills, different disciplines. You get people that are expert in structural and imaging, in the clearing and the labelling of these neurons. You can see the individual neurons now. And now we can put all this together and actually make a three-dimensional render of that piece of the brain. And that's just, this would have required, this can be done in a day now. This would have been months or years of work before. So we're hoping, you're going to get sick looking at this. This is, what we're actually doing is taking pictures of this brain. We rotate the brain, because optics work better in the XY plane that you're imaging. They're terrible in the depth plane. So to overcome that, you rotate the image and look at it from different, and then you've got to put all those pieces back together. So the team has been coming up with algorithms of how to assemble all these different views into a single coherent structure. And we're using some of the same technology that they use in the Xbox Kinect to map three-dimensional surfaces. You essentially, and even the way the new iPhone has got that camera that projects a bunch of dots onto your face, and these dots are called point clouds. And there's really cool software where you can look to see how these clouds of dots, because how else do you look at a three-dimensional surface? It's really hard if you've just got a complete surface, but by decorating it with dots, you can compare one pattern of dots to another pattern of dots and find a match, the best match of those dots, and that can happen essentially in real time. So I'll stop it there. So we're hoping that this is going to give us, because as I said, some of these epilepsies, we've been blind to the structural changes and what happens over time. You've been having epilepsy or other disorders. We're totally blind to a lot of these. So with these methods now, we can start to rapidly ascertain exactly what's happening at the structural level, and then even understand whether the therapies we're delivering are also rescuing or sparing the structural damages that occur as a result of these disorders. So I just want to leave you with that idea that, now more than ever, science is hugely interdisciplinary. It's normal to have computer people and engineering people, and mathematicians, clinicians and biologists all working in the same group. And I think that's changed, in the time I've been a scientist, that's changed more than anything. You see some departments now, the old traditional departments of physiology and pharmacology and anatomy don't really mean much anymore in the context of research. They still mean stuff in the context of teaching, but in the context of teaching, that's changing a lot, and it's very exciting to see this, and I think it's the only way we're going to move forward. So I hope I've given you a little bit of a feel about the way we're tackling some of these problems, and some of the progress I think is happening here at the Institute is really quite remarkable, and we're leading the world in some of the things that we're doing. So I just want to leave you with that. I think there's a lot of hope. The parents that we talk to, and we deal with a lot of parent groups in Australia and around the world. And, it's not all about doom and gloom. It's a horrible hand you may have been dealt. But I think, there is signs now for the first time that we can really change the lives of these kids. So thank you for attending, and I'm happy to answer any questions.